This is the University of Otago.
For those who don't know, my name's Peter Joyce. I'm Dean, University of Otago, Christchurch. And it's my pleasure to introduce our 7th public health lecture for the year, the 7th and last one. And we keep the best till last. This year, all 7 of the public lectures have also been inaugural professorial lectures, which are traditions that universities have, usually shortly after someone being appointed or promoted to professor giving this lecture about their work. So tonight it's my great pleasure to introduce Professor Mark Hampton. Now Mark did a Master of Science through University of Canterbury and then did a PhD here with Professor Christine Winterborn. He did postdocs at the Karolinska Institute in Sweden, and then he went to Harvard in the United States. And he's been on our staff since 1999. And he was promoted to professor at the end of last year, which officially took effect from the 1st of February this year. So it's a great pleasure to introduce Mark Hampton, who's going to talk about life and death under the microscope. Mark.
Thank you, Peter, and thank you everyone for coming along tonight. It's quite strange looking out into the audience and seeing a lot of familiar faces, but not in this environment that I'd expect to be here in this lecture theatre. What I want to do over the next hour is give you a brief, rapid overview of some of the research projects I've been involved with throughout my career. Because of time, in some places I'll go quite quickly and may not explain things very well or may lose a few people, but I guess what I want you to get out of the whole experience is to go away thinking or seeing why I wanted to do some of the work that I do and why it's really exciting and why science is fun and cool. If that doesn't work for you, my son Theo's sitting down here. He's brought his iPod. He told me if things get a bit boring, so you're welcome to come and see him. I also hope my voice is going to hold out as well. I've had a few problems with it for the last week or so, but we'll see how that goes. Okay, so I'm a cell biologist. I like to study cells, and cells are the most fundamental form or basic unit of life is how they're described. Life forms can consist of one cell, in the case of this amoeba sitting in the pond, or it's many cells. And for multicellular organisms, life gets a lot more complicated than that amoeba, in that they all have to work together to carry out the function of that particular organism. As an example, I put up humans. We have something of the order of 50 trillion cells. That's an estimate. It's very hard to measure or count that number. And there are over 200 different cell types, very specialised cell types that work together. They can't survive by their own. They work together to enable us to do all the things that we do. In fact, I think it's quite neat to think that these cells can collect themselves together in an organism that is sufficiently organised and intelligent to be able to invent a microscope and then look back down at the same cells that created them. Okay, I'm very interested in cell death. For that single-celled amoeba, death means end of the organism. For multicellular organisms like ourselves, it's a very different situation. And in fact, cell death is an essential part of life as a multicellular organism. I've put up there that over the next hour, about 5 billion cells in your body will actually die. It's not a bad thing, so don't worry. The other interesting thing is they won't just die, but they'll initiate this self-destruct mechanism inside them and intentionally kill themselves for the benefit of the host. I'm going to get on to that process a little bit later on, but first I wanted to introduce you to my favourite cell. This is a phagocyte, or in this case a phagocytic white blood cell. And its job in our body is to hunt down invading microbes, to ingest them and kill them. So you can see on this particular slide, this one's reached out an arm for this wee green bacterium down there, is about to grab and ingest that particular bacterium. The function of phagocytic cells was discovered by a Russian zoologist, Metchnikoff. It's an artist impression of theirs sitting looking down his microscope. The legend has it, and I'm not sure exactly how correct this was, that he performed this work while on holiday with his family in Messina, which is in Italy on the Mediterranean coast. And he was interested, or he was looking at these transparent starfish larvae under his microscope. And you can read his thoughts. This comes from his journal here. When a new thought suddenly flashed across my brain, and he was looking at these mobile cells within the starfish larva. And he had this thought that maybe these cells might play a role in the defense of organisms against intruders. At that point, he rushed outside. He went to the nearest rose, snapped off a wee thorn, came back inside, poked that into a starfish, and came back a day later to see what had happened. And again, that's an artist's impression down here of the transparent larvae, the starfish thorn there, and these mobile cells collecting around the thorn. I like that story for two reasons. One is there's that sort of eureka moment that we all have in science, probably not the same magnitude or significance as Metchnikoff or Newton and his apple or Archimedes in the bathtub, but it's a great moment, and it's the things that keep us going in the lab when all else, dealing with sort of the frustration and some of the more mundane day-to-day things. The second part I like about this particular thing is that his wife actually let him take his microscope with him on holiday down into the Mediterranean and took the kids out to the circus while he got to have a play with it. I think that's priceless. Okay, so as I said, it actually took quite a while for Metchnikoff's theory to become more widely accepted. He showed this in starfish, but then subsequently in a number of other organisms. And I like this wee movie here off the internet, and this is again now looking at our own blood cells here. This is a neutrophil on a glass plate surrounded by some red blood cells. And this really illustrates this white blood cell chasing this bacterium across the slide. It's following it by the chemical signals that are coming from the bacteria itself. So it's effectively sniffing the bacteria, pushing aside the red blood cells, grabbing that bacterium and swallowing. So this is a more detailed image of a similar process. So this is an electron micrograph where he's actually taken a cross-section through that white blood cell here. So this is the cell. This is its nucleus here. And these are a number of bacteria that have been ingested. So several bacteria here that have been ingested into their neutrophil inside these compartments, which we actually call phagosomes. So Metchnikoff won the Nobel Prize in 1908 for his discovery of this theory of phagocytosis. And he noted around about the same time as that he really didn't know how these things were killing bacteria at all. And that was something that he thought was the next thing that needed to be sorted out. That question is actually what I did my PhD on with Christine Winterborn and Tony Kettle here in the laboratory. And in fact, it's a question that we're still asking in the laboratory today, is that a number of the details, exactly what's going on, haven't been worked out. But what I want to do tonight is to describe one of the mechanisms. And I guess the first answer should be that there are multiple mechanisms, and that makes a lot of sense, that there are multiple mechanisms. If there was just one, it would be quite easy for a bacteria to be able to come up with a defense mechanism and to be able to become resistant to that. There are a number of mechanisms. The one that we study most within our laboratory is focused on the use of electrons to kill the bacteria. So the guy that was standing next to Metchnikoff when he got his Nobel Prize in Stockholm in 1908 would have been particularly interested in this mechanism. That guy's name, as you would have recognised from the photo, was Rutherford. And I have to admit to this point as being having a bit of an obsession with Nobel Prizes. To be honest, I'd quite like to win one. That would be great. But even as a young kid, I knew about Rutherford and his story and what was going on. I guess, well, like all kids, the first thing I wanted to be was an All Black, obviously. But as a wheezy asthmatic boy running around fields in Nelson, I realised quite quickly that that was never going to happen. But I did know about the Nobel Prize, and I thought this would be really cool. And I knew Rutherford was also born in Nelson, and that maybe that would give me a bit more of a chance. So Rutherford, as you're well aware, his concept behind the work that he was working on that particular time was the structure of the atom. So what I've put up here is a description, it's an illustration of what was known at the time as Rutherford's atom. And that's where you have this central nucleus, where you've got protons and neutrons there, and these negatively charged electrons are spinning around outside the nucleus. So that model was quite one of the early models, and it's been updated a lot since then, and atoms don't really look like that. But for the purposes of the talk, this is just about what we need, except to point out that actually the electrons in their orbitals tend to go in pairs. And so an atom that has an unpaired electron is known as a free radical. When it's unpaired, it either wants to get rid of that electron or to grab another one. And that is involved in a number of chemical processes and can also cause problems in biology. And in biology, the most significant free radicals that we have are associated with oxygen. So oxygen itself can take 4 electrons and end up as water, which is a good thing. But if it takes those electrons individually, we end up with these other things, these free radicals generated along the way. So just having one electron gives you this compound called superoxide. It wants to grab another electron to become more stable, so it rips it off things and can cause problems. You go through here to hydrogen peroxide, which is actually not a free radical because it now has 2 more electrons which are paired up, but it still wants to get through to water and can take electrons off other biological molecules. So let's get back to the phagosome. So again, this is an electron micrograph of a neutrophil. This is a phagosome here with a bacteria that's either 2 bacteria or 1 bacteria that's just in the process of dividing. Now, if we zoom into that very small space inside the phagosome, this is where some pretty amazing chemistry goes on. It's because it is such a small and it's an enclosed space that it's taken people a long time to work out exactly what's going on there. And also the reason why we don't know exactly today what was happening inside that phagosome. But what we do know from a number of work over many years, including some of the stuff that was done in our laboratory, is that free radicals are generated inside that phagosome. So there is an enzyme complex here. It's called the NADPH oxidase, or more simply a yellow box here. And that passes electrons one at a time onto oxygen to generate superoxide. So you can see the potential here that these molecules may be causing damage. Superoxide gets another molecule actually from reacting with another superoxide itself. So it takes one and goes back to oxygen. And you get this molecule, hydrogen peroxide, which you would have heard about as being a potentially reactive compound that's good for turning your hair white if that's what you so desire. And there's another, but not particularly lethal in the amounts that it's generated inside the phagosome. There's another enzyme here that Tony Kettle has studied for many years. This is the enzyme myeloperoxidase, and it can actually take hydrogen peroxide, turn it into hypochlorous acid, which is the active ingredient of granola. So now you can start to see that we have a mechanism, a potentially quite potent bactericidal mechanism for getting rid of these bacteria. So my PhD was basically focused in on this very tiny part of biology inside the neutrophil there. And so I was looking at the rates that white blood cells kill bacteria, looking at putting in inhibitors of this enzyme and this particular enzyme and see what effect it had on bacteria, introducing other inhibitors or scavengers, sticking things onto the surface of the bacteria to get them inside to that phagosome, and really tease out some of the pathways going in there. As I said, it's an unfinished story, and some of the questions are still being continued to be asked today. within the laboratory. It's also a very important question still, because this is the critical interface of the immune system and bacteria. And there are actually bacteria that do survive here. So Tony presented a paper at lunchtime at Journal Club about a bacteria called Yersinia pestis, which is what causes the plague. That bacteria goes into these phagosomes quite nicely and survives. To be able to come up with new strategies for targeting those bacteria, we really need to know how they're how's the cell trying to kill them, how the bacteria are actually overcoming that. And so we can come up with sort of more strategic therapies. For measuring bacterial killing, just to give you a rough idea of the assays that I was using, you take your white blood cells, you add bacteria to them, you mix them up for a certain time, you take a sample out, put it onto an agar plate like this, and you count how many bacteria are sitting on that plate. And then with time, that drops down. It tells you how fast those bacteria are being killed. So it's quite a simple assay. And in fact would be a very good assay if you only ever had to look at one or two plates. Unfortunately, it took me a lot more to do this. So this is a photo from a typical week of my PhD. At the end of the week, after about 3 experiments, the plates sort of piled up at the weekend. And I was sitting there, this photo is 20 years old, sitting there dotting away each of these bacteria and counting them and tallying up the numbers. So illustration of some of the tedium associated with science. Maybe also an illustration of the passion that does keep you going. And plenty of time to think about the next experiments and plan that as well. A review article came out from my thesis, so we got together and wrote that, which I thought was quite nicely titled Inside the Phagosome. I was also thinking at this time, at the end of the PhD, it was probably time to escape the phagosome as well and see what else there was to have a look at. So I come back to this apoptosis pathway here. So this is the process that really caught my attention. This is the self-destruct mechanism. And it gets activated in cells whenever they need to be cleared. So whenever they become damaged or the body no longer wants them, the self-destruct pathway gets activated. This here is a normal cell. After it's become apoptotic, it shrinks. It sort of packages itself up in these wee bundles and enables neighbouring cells to actually swallow the apoptotic cell and clear it away. One of the amazing things is that it still keeps its integrity, so it doesn't start leaking things out into the surroundings and causing damage at all. So definitely a process that I was really interested in having a look at. There were 2 links, though, to my actual PhD itself. The first relates to the resolution of information. And this is a slide, I think it's very similar to the slide David Murdoch showed a few weeks ago, actually. This is looking at a cross-section of a lung where there's an infection. And all of these cells through here are neutrophils that rush in to clear away the bacteria. This is what happens during the infection. And then this is several hours later. It shows how effectively those neutrophils have actually been removed, disappear from that site, which is really, really important actually, because obviously bacteria can be a problem, but you've got these cells full of all these toxic chemicals that are causing damage. So it's absolutely, even more critical that you get rid of them in some way. The other thing at the time was that there were suggestions that free radicals were also involved in driving the apoptotic process. And I've shown here, this is the same enzyme that was on the neutrophils for the purpose of killing the bacteria. The thing that came through at that point was that this enzyme was actually present on all cells. And you'd have to ask the question, what was the point of that? Because a lot of cells don't take up bacteria and are not generating all these oxidants. But all cells have this particular oxidase here. And the other thing was that antioxidants in the system could block this process. And I was particularly intrigued by reports that came out in certain cancer cells that were resistant to undergoing the apoptosis process, that they had elevated levels of certain proteins, and one of these proteins was thought to be an antioxidant itself. And so that's really what got me interested. So in 1995, I sent off 2 emails. So one was to a group in the UK who was studying neutrophils in inflammation, resolution of inflammation. I still haven't heard back from those guys yet. But the other email I sent was to a group in Stockholm, and that was the group that, the only group in the world at that time that was studying the role of free radicals in apoptosis. That group was headed by Sten Arrhenius, and he said, yes, please come. And again, I think for someone who was a bit sort of obsessed with the Nobel Prize, Stockholm was the place to go. So this image, this is not the Karolinska Institute, for those of you who know Stockholm. This is the Old Town Gamla stan. It's actually taken from the top of City Hall, the tower at City Hall, which is where a lot of the Nobel ceremonies go on. That's me there inside the building. That's where the banquet is held every year. That wee podium is where the Nobel awardees get to deliver their after dinner speech. So I've got close. And I'm ready for next time. Okay, so a bit more about the apoptosis process itself. So what we've got here is some cells. They're sitting on a plastic dish, having a look down on top of them with the microscope. And a juicer of apoptosis has been added. And the idea of this movie is to illustrate, so this is a time-lapse movie, this has been sped up, but this is still only over a few minutes, that dramatic morphological events that go on. And I think it illustrates it better than that first one I showed you, that the cells basically collapse on themselves and bundle themselves up, ready to be eaten by neighbouring cells. Here's a movie that was made more recently, in fact, just over a week ago in our laboratory using some more modern equipment, imaging equipment, and it also includes, I'll set it going first, again, cells sitting on the plate. Also thrown in a compound that you'll start to see turn red. But at this stage, you see all of these cells sort of bundling themselves up, it's coming off the plate, and as it looked like in the first movie. Not sure how long this is going to go for, actually, but okay, so what's happening is you start to see a bit of red colour coming on in the cells. Now, what's actually happening here is these are the enzymes in the cell that are responsible for triggering this apoptotic pathway. So these proteins are called caspases. They chop up regulatory structural proteins of the cell and enables that bundling to occur. Just look in this region there, that you get this red colour coming on nicely, then all of a sudden, bang, it's rounded up and done. Okay, so studying apoptosis at that period in the sort of mid to late 1990s in Stockholm was really great, because that was really the time that we started to get an understanding. These caspases were discovered, and all the other processes involved in apoptosis were being discovered, so that was quite neat. One of the key findings that came from my work at that particular time, so this is actually looking at one of those caspases itself. So this is the thing, there's a wee peptide in here, it goes in and it gets chopped up. And in that previous movie, there was a wee red tag onto that peptide, so when it got chopped, the tag would come off. That was the red colour that you were seeing. But what I showed as part of my work was that there's a portion of these caspases, and it's this particular amino acid in the protein that's shown in yellow, showed that it was actually really sensitive to being inactivated by hydrogen peroxide. And it was at that point that it made me start thinking about the early studies with the neutrophils and how they generate all of these oxidants. How could they be undergoing the same sort of process as what happens in other cells? And in that lab, I did have the opportunity to sort of work with a PhD student at the time, so it was sort of my first taste of supervision, and was able to start having a look at some of these processes going on inside neutrophils as they were dying. And what we were able to show was that, yes, the cell death pathways within the neutrophil were different to other cells. And well, I've shown this one here. This came a bit later on, back in Christchurch at the time, but it's one of the best illustrations, I think, or certainly a nice visual illustration of what was happening here. And this, just to confuse you, I've changed the colours here. So there are actually 3 white blood cells on this particular slide, so I'll show 1, 2, and 3 there. Okay, you can see that. Now the red one has taken up bacteria. The bacteria have actually been coloured red in this example. So we have one neutrophil that's taken up red ones, and here are two alongside it that haven't eaten anything at all. Now the green stain now is the casposes. Okay, so here we're looking at neutrophils that have their casposes starting to come on, but the ones that have eaten the bacteria, the green has faded a lot, so there are no active casposes on in those particular cells. Quickly jump to today, and actually at the moment in the lab what we want to go and do, what I'd like to be able to do is actually go and reinvestigate some of these cell death pathways in the neutrophil again. The reason for that is, well, one, it's still important to know what's going on and to know how they're cleared, but a number of other death mechanisms have now been shown. And so I was at a cell death meeting a couple of weeks ago in the States, and apoptosis got barely a mention, and there were these other forms of cell death that were going on. And one in particular, I think, is potentially what is actually going on inside the neutrophil. So it'd be quite cool to go back and reinvestigate some of those other systems there. The other thing that we have been doing in the laboratory as well is looking more recently as sort of the regulatory role that these oxidants may play. So not just having a role inside the phagosome for killing bacteria, but it's been shown that they get generated elsewhere, or maybe not elsewhere, but actually can come out of that phagosome and do other things. And more recent work in the laboratory, again, has shown that there are particular proteins involved in the regulation of inflammation that the free radicals the neutrophils generate can interact with those proteins and change the function as well. So there's a lot more that we need to know about free radicals and inflammation, and it's not all just about what's going on inside that phagosome. Right, so in the second part of the talk I want to leave the neutrophil for now and go back and talk more generally about cell death pathways in other cells, tell you about mitochondria and put it in the context of cancer. And so in this particular one, this is just something, again, I've grabbed off the internet, and it's illustrating, well, this is a cell here, a membrane. Here's a nucleus. You won't be able to see it way down here, but this is the point where you get caspase activation. So there are a number of other processes leading to the point of actually activating these caspases to get the cells to die. And you'll be pleased to know I'm not going to take you through any of those. The sole reason for putting this slide up is that there's this strange-looking thing right here where all of the arrows seem to join to and come out of to link to the caspase activation. And that organelle is the mitochondria. So I've told you neutrophils are my favourite cell. The mitochondria is certainly my favourite region or organelle within a cell. This is an artist impression here of a mitochondria. Two mitochondria inside a cell, one intact, the other one has been sliced through the middle here. And you can see there are two membranes within this particular mitochondria. So what do these organelles do? Actually, before I even show you what they do, just to show you actually that was the artist's impression. In real life it's often not like that. These things are actually very dynamic, so they're constantly fusing with each other and budding off, and so you get these structures that look like tubules. So in green here, this is actually a real mitochondrial network inside cells. So what do they do? Okay, so Star Wars fans had an early inkling, if they were paying attention during the movies. And so, yeah, George Lucas nearly got it right. So he called them midichlorians, not mitochondria. So these microscopic life forms, and he said they communicated with the force. The real thing is they actually generate the force within the cells. So this wee guy, Anakin, who had a bit of an anger management problem, but he was shown to have 20,000 of these mitochondrions within within each of his cells, which gave him special powers. That 20,000 is actually quite a good ballpark. So our cells they vary in the number of mitochondria they have, but many of them can have 20,000 or even up to 100,000 mitochondria within them. And this is the place where basically our food gets turned into energy. And here's a sugar molecule here, glucose, which has been straightened out to make it easier to look at. We eat this. all of this process doesn't actually occur in the mitochondria. It can occur, the first part occurs outside, the second part occurs within the mitochondria itself. But essentially these bonds between these carbon atoms get pulled apart, and so the glucose ends up as carbon dioxide, and we get energy from that. Now I want to go into a bit more detail about the process itself, because it's, well, two things. It's quite important for what I want to tell you next, but I think it's also really important to actually understand how we generate energy within our body, how our own engines work. And so to illustrate that, I've thrown up a couple of systems here. One's the internal combustion engine, which many of you are going to leave here tonight, jump in your cars and drive home. And you've probably got some understanding here. So the fuel gets squirted into here, into the piston. There's oxygen there as well. The wee spark causes an explosion inside that due to the burning of the fuel. It's obviously not glucose, but octane, which is a bit longer, a couple of extra carbons. That pressure that builds up forces that piston, which moves the camshaft, is it? Anyway, the wheels turn around. I'm not a mechanic, okay? The other system I've shown here, another way of generating is generating electricity through a hydroelectric power dam. So we get water building up behind a dam that then flows downhill. Again, turns these wee turbines inside the dam that generates electricity, which then heads off to a household. So how do we do this process? How do we convert this to energy? And any of these processes similar to what might actually be going on inside our own bodies? The answer is yes, and it's actually both of them in some way. And I'll show you the detail shortly. Slightly different in the sense that we do burn that glucose that I showed you using oxygen to turn it into carbon dioxide, which gives quite an explosive energy, but we don't do it all in one go. We sort of do it in small fragments so things don't explode. But we have that process. We also do make use of this system here, but it's slightly back to front in that we use electricity within our cells to drive compounds, not water, but drive hydrogen ions or protons, I'll show you shortly, up a gradient. And then that gradient goes back downhill and spins a wee turbine inside our own mitochondria to generate ATP. And so this is a bit more detail of the process. So what we've done here is we've zoomed into a mitochondria. This is a cross-section of a mitochondria. I said it had two membranes. This is one membrane here. The second one you can't see. Imagine that's on the top there. At this point, so our glucose has been pulled apart. So the electrons that were within that bonds, that energy have been moved around. There's actually an intermediate takes them into here. But these electrons get fed into this wee complex and pass through a number of protein complexes all the way down a chain, exactly like electricity flows in a wire, down through here, and they end up on oxygen. And they turn oxygen into water. And as the process occurs, so as these electrons are flowing through here, what happens is you get these protons get pumped out. And so you form this gradient. So you have all of these protons on the outside of this membrane. Now, these protons can't pass through this membrane, but there's a certain pump within our cells where these protons can come back through and actually spin the pump, enable it to generate ATP. And I have to show this movie here because this is actually, it's a model of this pump in action. And effectively you get the opportunity to see really how your own internal engines are working. So you can't see the protons, but they're flowing back down through here. That's causing this protein complex to be spinning around. It has an awkward looking shape here, and it's causing movement. And these three things coming out here are ADP, and it's coming in and being turned into ATP on this particular pump. Oops. Okay, last Star Wars one, promise. I've put this in here just to illustrate or remind me to tell you that this rate of energy conversion that goes on inside mitochondria is amazing. It's actually 10,000 times the rate of the energy conversion that goes on inside the sun itself. Fortunately, they are quite a bit smaller than that, though. Another wee interesting tidbit for you trivia people is, where did these mitochondria come from? And again, that was the artist's impression. They actually look very similar to bacteria. And the answer is they actually are bacteria. And so this woman here, Lynn Margulis, proposed this so-called endosymbiont theory. And this was, and so it is a theory because we can't go back in time to work out whether this really happened, but all the evidence is there, is that there were, initially our cells didn't used to have mitochondria, and one of these phagocytic events, exactly like I showed you with the neutrophil, occurred one day where a specific type of bacteria was taken up that wasn't actually killed by the host cell. And so then those basically two organisms live together. The bacteria inside the cell generating the energy that the cell needed to become multicellular and do all of those other amazing things that cells do. Was very controversial at the time, but now it's almost widely accepted in the field that this is actually true. So again, that's quite something to think about, that we're actually an alliance of two independent life forms that have got together for this mutual benefit. The other thing is, behind every great woman there's a great man. I had to put this in here as well. So Lynn was married to the famous science popularizer Carl Sagan. And it was his TV series Cosmos in the early 1980s that really hooked me onto science, and watching this guy on TV, and some of the older members of the audience may be able to remember him and some of the quotes that I've put up there. Still have the DVD series at home. Unfortunately, the outdated special effects and some of the fashion sense doesn't seem to appeal to my kids or any other kids of today. But it'd be great if there was, someone else nowadays that could take his place. Okay, I've gone off on a tangent. Sorry about that. But I did warn you, I wanted to tell you why I find some of these things interesting. Right, so back inside the mitochondria. Those of you who are taking close notice might have realised there's a potential for real disaster here. And there is a real overlap with what happens in the neutrophil. And this is where we have electrons wandering around, going on to oxygen, and the potential to generate, rather than to be converted into water, but generate these other free radicals within the cell. And this particular enzyme that actually operates here, where the electrons are fed one at a time onto oxygen to give water, works really well. There is no leakage of free radicals there. But off this electron transport chain here, as these electrons are flowing through, they can jump onto oxygen. So it's well known that you do generate superoxide inside all mitochondria, and that can go to form hydrogen peroxide as well, and that can be damaging. And in fact, it's been linked to a number of diseases, neurodegenerative diseases especially, and also to be associated with ageing. That this free radical generation does cause damage. The other key thing for the purpose of my talk, and why we actually started talking about mitochondria in the first place, is actually also it's the key site where this apoptotic self-destruct program gets switched on. And so I've just illustrated this here. This is supposed to be the outer membrane here, that when a cell becomes apoptotic, a signal goes that forms a pore into this membrane, and things get leaked out of here. In this case, it's one of the proteins that's actually involved in shuttling electrons, comes out, goes into the cell, and this is what activates those caspases, those things that turn cells red. And so a very important part of the process. And it's also the working hypothesis of many is that the free radicals that are generated during this particular process are involved in the initiation of apoptosis. So again, a positive example of what free radicals can do within a cell. I should probably qualify that. Positive depending on whether it's good or bad if you want a cell to die. So in some situations we don't want cells to die, and this happening is going to be a bad thing. But in other cases we do want cells to die. That's where the cancer story comes into play. And so it's been well recognised now for quite a while is that cancer cells don't die when they should. They have switched off these self-destruct mechanisms, and so that actually enables them to grow in places that they shouldn't be growing, where they're getting signals that you shouldn't be here. Also makes them very hard to treat as well. So a lot of the chemotherapy and radiation treatment is attempting, actually activates these self-destruct mechanisms. And so if you've got a cancer cell where these have been turned off, this obviously makes that quite difficult. So the idea of a number of people is maybe we can switch back on these self-destruct mechanisms and encourage cells to die. And one of the things that we're looking at is maybe we could target the antioxidant systems within the mitochondria to get a few more free radicals being generated inside those mitochondria of the cells we want to kill to be able to promote cell death. The important thing here is that we want to be trying to kill cancer cells as there's something different about cancer cells. And this is actually, there is evidence they are different, that their pathways are set up quite differently, that metabolism is different in a cancer cell from a normal cell. So this is a German biochemist, Otto Warburg. This is actually a real photo, and if there's ever going to be a movie made about this guy, I think you could recognise who the actor might be. But this guy's no Mr. Bean at all. He's the guy that, the cytochrome oxidase that I talked to you about, he won a Nobel Prize in 1932. He was the guy that discovered the cytochrome oxidase, the things that chucks the electrons onto oxygen to generate water within the mitochondria themselves. And for those of us that get a bit disappointed that no one seems to listen to any of his ideas, this is an interesting timeline here. His theory was first, so it's German, it was first published in 1956, and no one took any notice of it for 40 or 50 years, well after he died. But just in the last 4 or 5 years, it's really taken off. So there have been, last year, 258 papers actually specifically talking about his effect, and it's probably one of the hottest areas in cancer biology now as to looking at metabolism in cancer cells. Okay, and for those of you like me who are going to spend a number of weeks this year writing grant applications, this is Warburg's grant application from 1921, so he wasn't even famous, he didn't even have his Nobel Prize at that point. Basically it says, I require 10,000 marks, and he got them. Okay, so just to finish the last sort of sciencey slide I had is again just bring this back to some of the things that we're doing in the laboratory at the moment with around this mitochondria oxidative stress cell death and cancer thing. I've put 3 different things up here on this particular slide. So we're really interested in understanding the antioxidant systems of mitochondria, or in fact of cells in general, and how are they working to remove the free radicals that are generated within cells. This is a crystal structural model, of my favourite protein. This is a peroxyredoxin. Its job is it removes hydrogen peroxide that's generated in cells, but it has these really cool structures, and I'm only showing one here of a donut type shape, but these things can stack on each other, they can form these interlocking rings, and one of the things that we're interested in is these things not only seem to be very good at removing peroxides, but also interacting with other proteins in the cell to tell the cell that it's actually under stress, that there are more things being generated, and give the cell a chance to adapt to that in some way. So we're trying to understand how these proteins work within the cell. The other thing we're trying to do is looking at cells that are actually dying, undergoing various types of cell death, and trying to understand the changes in the free radical generation and the antioxidant pathways within those particular cells. Again, trying to understand that. Obviously, the ideal thing would be compounds that we could use that could promote stress and promote cell death. We have a number of projects looking at this particular class of compound. These are called isothiocyanates. These are actually naturally occurring compounds in cruciferous vegetables that are responsible for that pungent odour and bitter taste of some of these things that people don't like to eat, like the watercresses and the broccolis and wasabis. And we've shown that these compounds here are very good at killing cancer cells, even cells that are actually, some cells that are resistant to undergoing apoptosis, and trying to understand exactly how they're working. And unfortunately, you aren't able to get enough of these things in your diet to be able to use them as a therapeutic. But we're trying to understand, look at a range of different compounds, how they're acting inside cells, and maybe we can use them to promote cell death in cancer cells. Okay, right, so just wanted to finish off with a few acknowledgements. And I'm going to start with my family to start with. And obviously I'm very passionate about my work, but for me my family is still number one and always will be. And so to, there's dotted up there and different things. So to my wife, Anne-Marie, yeah, we've been on a lot of fun journeys, and both before and after the kids, and faced a few challenges on the way, but we make a great team, and we've got through them all so far. So thank you very much for that, and yeah, and I promise never to bring my microscope on a holiday. Thanks also to the bigger extended family, especially my parents who are here tonight, for the unconditional support that they've given me over many, many years, and always making me feel like I could achieve anything if I set my mind to it. To my colleagues, it's actually been 25 years, so there's many of them, too many to list, actually. But as I said to our group members on Friday night, where we had a wee function, over those 25 years, I've been very fortunate to be able to visit some nice places with and nice or good universities in some nice towns, but feel even more fortunate to be able to have actually grown up in this particular building. So thank you very much for making the University of Otago Christchurch a home for me, and something I've always wanted to return to. Now I have to do this and zoom in on 4 special people on this particular image. I don't know when that group photo was. I'd been here a couple of years, so I think it's the early 1990s, and some of you may recognise yourselves there, but I had to zoom in on 4 for, of course, the sole purpose to illustrate the effects of free radicals and ageing, maybe. No, it's not. So Tony and Marguerite, you guys have always been my scientific heroes and my heroes, full stop, and so I've been very proud to walk in your footsteps, and thank you for everything. Christine, as you know, I'm not very good at this sort of thing at the best of times, and actually, yeah, I don't know, I think it's hard for anyone to come up with the right words to thank someone who's given me the opportunity to actually do something that, spend my life doing something that I really love. And so, yeah, thank you doesn't seem to be the right word or the right words, but they're the only ones I can think of at the moment. So thank you very much for that. And thank you all for listening.